Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year 1977, Frederick Sanger, a name now synonymous with groundbreaking advancements in molecular biology, published a seminal paper DNA sequencing. This work, a culmination of persistent investigation and intellectual curiosity, revolutionized our capacity to decipher the intricate code of life. Its genesis lay not in a vacuum, but within a fertile intellectual soil cultivated by preceding discoveries and the persistent pursuit of understanding the very essence of heredity. The intellectual landscape preceding Sanger's breakthrough was dominated by the blossoming field of molecular biology. The elucidation of the double helix. Watson and Crick in 1953 had unveiled the structural blueprint of DNA, but the precise sequence of the nucleotide basidine, guanine, cytosine, and thymine remained a formidable enigma. Early attempts at sequencing, primarily focused on RNA, and proven cumbersome, leading to the development of comparatively inefficient techniques. Sanger, a two-time Nobel laureate, possessed the keen insight to perceive the limitations of these methods and the audacious ambition to conceive something entirely new. His prior work, particularly his successful sequencing of insulin in the 1950s, demonstrated his expertise in protein sequencing. This experience, especially his understanding of how to chemically cleave proteins into smaller, manageable fragments, proved to be invaluable. Sanger's approach, developed with the crucial collaboration of Alan Coulson and Steve Nicklin, centered on a novel strategy, the chain termination method. This ingeniously simple yet remarkably effective technique, often referred to as the Sanger method, harnesses a set of modified nucleotide building blocks, specifically deoxynucleotides. These deoxynucleotides, lacking a hydroxyl group at the 3' carbon of the deoxyribose sugar, function as chain terminators when incorporated into a growing DNA strand by a DNA polymerase enzyme that prevent the addition of further nucleotides, consequently halting the synthesis. The process involves replicating a single-stranded DNA template, referred to as the unknown sequence. 4 separate reactions are performed. Each reaction contains the template DNA, a primer complementary to a known region of the template, a DNA polymerase enzyme, and a mixture of the 4 regular deoxynucleotide triphosphates: deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, and thymidine triphosphate. Crucially, each reaction also includes a small amount of a single deoxynucleotide, distinct for each reaction. For example, one reaction would contain deoxyadenosine triphosphate, another deoxyguanosine triphosphate, and so, as the DNA polymerase synthesizes a new strand complementary to the template, the incorporation of the deoxynucleotide, which occurs randomly, creates a set of DNA fragments of varying lengths. Each fragment terminates at the position where the specific deoxynucleotide was incorporated. The resulting fragments are then separated by gel electrophoresis, typically polyacrylamide gel electrophoresis, a process that separates molecules based on size. The smaller fragments migrate most rapidly. Hence the size of an individual fragment reflects the sequence on the template strand. The gel would allow separating individual fragments based on their size, with the shortest fragments migrating furthest and the longest fragments the least. The final step involves visualizing the fragments. The gel is stained, most usually with a fluorescent dye, so that the bands can be seen under ultraviolet light. The positions of the bands reveal the sequence of the DNA. The sequence is read by starting with the smallest fragments at the bottom and moving towards the larger fragments, allowing researchers to deduce the order of the bases in the original DNA template. The sequence is read by examining the bands and comparing the gel lanes for each of the 4 deoxynuclei. The elegance of the Sanger method lies in its ability to provide a high degree of accuracy when correctly executed. Moreover, the method is relatively straightforward, enabling it to be adapted fairly readily. The technique offered a significant improvement over earlier methods in both speed and precision. This methodology provided a revolutionary leap forward in the ability of researchers to decipher genetic code. In essence, Sanger's method transformed the task of DNA sequencing from a laborious endeavor into a manageable, widely accessible undertaking. The immediate impact of Sanger's DNA sequencing method was profound. The technique offered a crucial toolkit for scientists delving into the mysteries of the genome. As the technique allowed researchers to readily determine the order of base pairs, it was instrumental in the analysis of a variety of projects. The technique was immediately useful in the study of viruses and bacteria, the development of novel diagnostic tests understanding the mutations related to. One of the earliest and most striking applications involved the complete sequencing of the genome of bacteriophage 5X174, a small virus. This accomplishment, completed shortly after the publication of Sanja's method, marked the first complete DNA genome to be sequenced. This achievement dramatically accelerated the growth of the field of molecular biology. It set in motion a cascade of other applications. The Sanja method, or, as it eventually was called, traditional Sanja sequencing, became an industry standard for several decades. It provided the foundation upon which all subsequent forms of sequencing developed. The method's versatility supported its lasting significance. The method could be adapted for a large degree of projects, from identifying genetic variations to understanding the evolution of species. The Sanja method was a workhorse for research. As technology advanced, the Sanja method underwent continuous refinement. Automation was a key area of evolution. Automated sequencers emerged, which significantly reduced the time and labor required for DNA sequencing. These machines employed fluorescently-labeled deoxynucleotides, allowing the different fragments, terminated at the 4 different nucleotide bases, to be distinguished by their color. This led to a one-lane separation on the gel. The data was digitized, enhancing the speed of the process. The reception of Sanja's work was, unsurprisingly, enthusiastic. The scientific community recognized its transformative potential from the very beginning. The method received widespread acclaim, and it rapidly became an indispensable tool for researchers across the biological sciences. The method's accessibility and utility made it a fundamental component of countless research endeavors. It facilitated the elucidation of genetic codes, the investigation of disease mechanisms, the advancement of evolutionary critiques, while present, pertained more to the method's limitations. For longer sequences, the method can be complex and time-consuming. Furthermore, the analysis of the data required skilled interpretation, and the method, while robust, was still subject to errors. These concerns, however, did not diminish the overall significance of the method. Instead, they stimulated efforts to improve the technique. The Sanja method, however, was not without its limitations. The expense of the necessary equipment and reagents was a significant consideration. Furthermore, the method's capacity was necessarily finite. The method could not match the throughput levels of more advanced technologies. These limitations provided the catalyst for the development of second-generation sequencing. These new technologies, often referred to as next-generation sequencing, offered drastically increased throughput, enabling the rapid sequencing of entire genomes. These new approaches, though based on different methodologies, also relied on Sanja sequencing in their development and validation. The influence of Sanja's DNA sequencing method extends far beyond the realm of pure scientific inquiry. It has profoundly affected human health, agriculture, and forensics. For example, in the realm of medicine, the Sanja method has been instrumental in the identification of genetic mutations associated with diseases, such as cystic fibrosis and Huntington's disease. This information has facilitated the development of diagnostic tests, enabling early detection and personalized treatment strategies. In agriculture, the method has aided in the improvement of crop yields and the development of genetically modified organisms. The same technique finds applications in the criminal justice system. The individuals whose prior work paved the way for Sanja's accomplishment were several: Oswald Avery and his colleagues' identification of DNA, James Watson and Francis Crick, who delineated the double helix structure. Moreover, Sanja himself drew inspiration from his earlier work on protein sequencing. The knowledge he gained was directly applied to this work. The influence of Sanja's work is undeniable. The method inspired a multitude of scientists to explore the genetic code, and the work has driven the development of sequencing technologies. The method's impact will be present for years to come. The Sanja method is a cornerstone of genetic research. The method is not only a testament to the power of intellectual curiosity, but also a reminder of the vital role that scientific inquiry contributes to human progress. Sanja's DNA sequencing method remains a symbol of scientific innovation. It has transformed our understanding. Our technique is still relevant in months.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.